So in my talk today, I really want to go into more of the systems that we have. So, kind of highlight some of the technical capabilities, as well as some of the applications that might not have been mentioned today. I have to say that Professor Hoffman did a very good job and cut a couple of my slides, yeah. So, and in general, as we already know, we are a microfluidics company in Sweden, and we are really focused on the flow of controlling the flow of liquids in small volumes. So here on the left, you see kind of our chip designs. So this is kind of a chip PDMS based for Biopen, as well as for BioPixlar bioprinter, and a glass chip for Dynaflow. So Edward already showed the portfolio of our products. As I mentioned, I just want to quickly go through each of them to kind of highlight the technical features and show some of the maybe other applications. As I mentioned, Professor Hoffman did a fantastic job about introducing the Biopen as a system. I don't want to add too much to that introduction, other than show maybe from a different perspective how the Biopen works. So here we have kind of a mathematical model of that imaging of that bubble, right? So as mentioned, the Biopen has 3 channels at the end. It has a middle channel for which the solution is pushed out, and the 2 channels next to it are kind of a vacuum channels that are absorbing the solution back into the system. And this creates a microenvironment from where there is basically no dissolution into the surrounding areas. So one of the concepts you see here, you can really encapsulate a single cell in that microenvironment. It's very flexible. So the one of the main points of the Biopen is really the control of what you want to target when. So the timing, also mentioned in the previous talk, is you can have short pulses or really prolonged solution delivery to a particular cell. And since you have multiple channels, what is that you deliver at what time. From the application areas, it's a very broad application area that we have for Biopen. Really just some of the studies in single cells. Obviously that's one of the main application areas that we have. In this case, studying single cells, or more specifically the retraction of individual the retraction of the individual cells of a cell protrusion. So the Biopen was kind of used to really target a protrusion and then induce a retraction in this cell protrusion. Another area also going to the effects of how the Biopen works and is designed is cell-to-cell molecular transport. In this case, targeting a single cell in a network, and then basically studying the transport between the cells, so in intercellular transport. The same concept can, for example, be used in neurosciences, where you can target a single cell in a network and observe network-wide effects. A slide that was shown by Professor Hoffman, and actually the study published by Professor Jedermark, who will go on next. Here, I just want to add that the Biopen is also very from this, it's very adaptable in a way that it can work with additional probes together. So, for example, in electrophysiology, the Biopen can work with the pipette together to kind of deliver the solution in combination with electrophysiology recording. And another application, the new one, a bit more exotic, in mosquito neurobiology, a group from Rockefeller University studying mosquitoes. They used Biopen to deliver whole blood to the tip of the mosquito blood feeding needle. And they were interested in recording the neuronal responses in mosquito after delivering the blood. And last in the Biopen part, it's another area that was mentioned. So the Biopen also collects from the environment. So not only the delivery, but the Biopen can also collect kind of interesting debris. But in this case, this was a surface adhered vesicles, where they basically use the Biopen to actually collect these vesicles inside the kind of waste channels or specific channels, and then extracted this collection and further analyze these lipid vesicles. So much I wanted to add about Biopen. Shortly, I also want to show about the BioPixlar. This will definitely cut into some of the Gavin's talk, but I really have just 2 slides. Here, what we have done is really the point is we have expanded the concept of Biopen by just delivering the solutions in a very precise way to actually deliver cell suspensions through the same principles that apply in Biopen. So with recirculation flow, we can now have cell suspension, deliver cells in a very precise way down to a single cell level into the defined area, into the in vitro cell dish. So some of the capabilities of bioprinting. So we really can go from placing a single cell into where you want it to position in the dish, to different combinations of multicellular 2D models up to a 3D modeling. The BioPixlar technology is really for high resolution printing, where you want something printed with very precise, in a very precise way, and basically down to a single cell level. And last but not least, another compound delivery system, DynaFlow, specifically designed for ion channel screening. So in difference to Biopen, this has a much wider, a much more solution delivery capability. So it has a 16 channel, 16 solutions. It has 16 channel. It is connected, as you see here, with a patch clamp, manual patch clamp. And it can also switch much faster between different channels. Another image here. So it's a glass chip with 16 solutions. Similarly to what Biopen, it has a very low compound consumption. It has a very fast solution rate, because what happens is that you place a cell next to 16 channels that are flowing out in a laminar flow, and then the stage switches between each channels. And I think this is something that will also be described in more details with Professor Jedermark. And yeah, just an image how this looks like in under the microscope. So you see here is a cell on a patch clamp, on a patch pipette, placed directly in front of one of the 16 channels. So the idea is that what happens is that you have laminar flow from all of the 16 channels. So the cell in this area is always surrounded from the fluid from the specific channel. And the solution exchange happens by simply switching the chip to the next channel. So the cell stays always at this particular place, and the chip moves to another channel. So this enables very fast solution exchange between channels, and is specifically for electrophysiology applications. And with that, I really want to finish my talk here. So as I said, the point for me was really to kind of show all the capabilities of the systems. And yeah, I'm open for a couple of questions, or otherwise give the floor to our next presenter.
Thank you very much, Matija. Is there any question from? Yes, there is one. Perfect. So the question is the flow unidirectional? So I don't know if you are talking about the DynaFlow, the Biopen. Okay, so are you talking, so Dr. Padmanabhan, are you talking about the Biopen? Okay, so with the Biopen, is the flow unidirectional?
sorry, can you specify in what is unidirectional for you?
He will be typing. He must type.
Yeah, I see. So you showed there. Yes, there in Biopen there is a recirculation fluid. So the flow is it goes from one channel out and basically into the two channels next to it back in.
So
I'll
if I may
jump in. Sure, Gavin.
So yes, it's essentially unidirectional. By recirculating, what they are describing is the fluid exits one channel and enters another channel, but the same fluid is not reused. It essentially only ever goes one way. So from the delivery channel, it goes out, and from the aspiration channels, it goes in. But so effectively, you generate what's called a hydrodynamically confined zone, where a liquid is always moving and being delivered. But the solution which is collected back isn't re-delivered. That collects back into a separate internal waste within the device, so that you don't contaminate any other cells in your sample.
So okay. So the second question is, what are the advantages of the Biopen versus competitors? So
I think the main advantages is what already Professor Hoffman described. It's really targeting very precisely inside the in vitro cell culture, right? So it's targeting single cells or small group of cells, very small areas, without kind of interfering with other cells in that area. And another thing that is also very important is a low level of consumption of compounds that is being used, because what we are talking about here, it's flow in nanoliters per second, and as mentioned, right, the maximum volume, it's actually 35 microliters. So I think these are the two main points for the Biopen.
okay, thank you, Matija. We have another question. I'm sorry. Could Biopen be used for cartilage tissue repair? So I think there must be a confusion. Maybe it's the BioPixlar, right, that we are talking.
Yeah, I mean, it depends on the application, right? So it depends what is the exact application. In general, if it's a cell delivery to something, it would be the BioPixlar, of course, right? And also with that, we would need to discuss in more specific what exactly would you want to do with this particular application. So what exactly is to be delivered and how, basically. And we are happy to take this discussion after the meeting. So I think it's in general it's very hard to answer.
Okay, thank you, Matija. So we will connect with Dr. Dudic afterwards. And there is another question then. Do you know anything about the effect of turbulent flow on mechanosensitive ion channels endogenously expressed in test systems?
Gavin. No, because we had this discussion yesterday. So
we have had this discussion recently. So it's actually very difficult to look at effectively the shear stress which is imparted on cells attached to a surface. I mean, there's actually very many components to it. I mean, one thing that we do have is that we, the hydrodynamically confined flow is actually within a laminar kind of stream environment as opposed to a turbulent flow, where we're not at the kind of size scale or the flow rate where you would actually get turbulence. We're in a laminar stream. And what that actually kind of leads to is that the flow which is actually being exposed to the cell, the last couple of microns, actually where the cell is sitting, is actually a very small cross section to the fluid flow. And as Professor Hoffman was stating when they were describing their experience on the solution exchange, it's such a small volumetric flow rate, which actually the cell, or the cross section of the cell feels, that the simulated stress on the cell is on the order of about 8 piconewtons. Now, we've tried to stress activate certain ion channels to see if we can actually experimentally measure what stress will be done. So we've run experiments on astrocytes and things, other cells which actually do show stresses upon local fluid exchange, and we haven't successfully been able to determine what our actual shearing is, but we haven't been able to stress activate any ion channels to date. Now, we don't try to purposefully do that, so I imagine if we sit right over the top and directly flow all of the flow over one cell, that we probably can start to increase the shearing. But as far as inadvertently activating mechanosensitive channels, we haven't been able to do it. And no one of our customers that does ion channel work has described any, let's say, negative effects of the flow on the cells.
Okay, thank you very much, Gavin, for this answer. I hope that answer your question, Dr. Mule. Okay, so now if there is there any other question, or everyone is good? One pop up right now. Okay, so we have a new one from Dr. Kuckley. When you bring the Biopen close to the cell in cell culture or in the slice, how do you prevent that bath solution / culture medium enters into the pen by capillary forces?
I can answer quickly. So what we actually do want the bath solution to enter the Biopen. That's just to clarify, we do want that to occur. We're actually introducing one, let's say, liquid compound into another liquid environment. So we're just overcoming any diffusion by maintaining this hydrodynamically confined flow region. So by continuously flowing in a laminar environment, they don't mix, but you can introduce one into another. The bath solution, as you accurately call it, is taken up into the device, but that goes into a waste region of the device. The solutions that you're trying to deliver and the solutions that are in the bath never, let's say, mix. They all waste goes to internal waste wells. So effectively, you have two wells in the chip, which would then mostly contain your bath solution, because that's what's being aspirated mainly from those two side channels.